Rab13 and its role in cancer regulation. Rab13 is a small GTPase and has many implications in current research due to its effects on the progression of various types of cancer. A GTPase is a G protein that acts like a molecular switch in signal transduction. It has on and off states. This family of enzymes participate in the hydrolysis reactions of GTP, and this occurs at a highly conserved G domain for all GTPases. GTPases are active when bound to GTP and inactive when bound to GDP. Guanine exchange factors promote activation by effectively switching out GDP for GTP, and GTPase activation proteins promote inactivation.
Rab proteins are a family of around 70 proteins in humans associated with all major steps of vesicle trafficking. Each Rab is concentrated in specific vesicles or intracellular organelles, as evident in the image. This includes the synthesis of membrane vesicles and tubules loaded with cargo, transport of these molecules on the cytoskeleton, and lastly, cargo delivery.
Rab13 controls many cellular functions which are changed in cancer cells. Rab13's trafficking pathways give rise to tumors, and it is referred to as a significant driver in cancer progression.
In cell metabolism, Rab13 translocates the glucose transporter called glucose transporter type 4, or GLUT4, to the cell surface in response to insulin. The insulin exposure leads to phosphorylation that inactivates the GAP, which in turn increases the concentration of active Rab13. This leads to both an increased sensitivity to glucose and tumor growth.
The formation of tight junctions is important for adhesion in epithelial and endothelial cells. In addition, phosphorylation of VASP allows it to relocate to tight junctions to establish adhesion. Rab13 can inhibit this pathway by regulating pKa, which disrupts the phosphorylation of VASP. As a result, VASP can no longer associate with intracellular junctions, and the integrity of tight junctions are lost.
Rab13 is also involved in cell migration and scattering. Once activated, it recruits actin-binding proteins to cellular protrusions. The protein MST1 can also phosphorylate VASP and lead to Rab13 activation. As a result, integrins are delivered to the cell surface and the actin cytoskeleton is rearranged, leading to cell migration.
These are example pathways that lead to cancer progression. Rab13 increases GLUT4 levels, which leads to increased glucose sensitivity and tumor growth. This protein also weakens the tight junctions of epithelial and endothelial cells, allowing cancer cells to spread easily. Other studies suggest that Rab13 mRNA accumulate in membrane protrusions to increase metastasis.
Rab13 is significantly upregulated in the majority of cancers, and its expression correlates with cancer progression. A common gene of study related to cancer is p53, which promotes Rab13 as a transcriptional target. Gain-of-function mutations in p53 upregulate genes that are associated with cancer progression, including Rab13. Other factors contribute to the upregulation of Rab13 in several cancers, including inflammation and repression of its inhibitors.
RNA from the cells were purified and analyzed by RT-qPCR. The level of each RNA was compared to the cell body fractions, and Rab13 mRNA had the highest localization ratio compared to other proteins to identify it as the highest in accumulation of protrusions. Results show that Rab13 regulates cancer progression by localizing its translation, forming protrusions, and promoting metastasis of cancer cells.
Another study was conducted to analyze what influences Rab13 mRNA localization. The 3' untranslated region of Rab13 was attached to a beta-globin mRNA with GFP fluorescence in live cells and monitored. The results showed that the beta-globin mRNA was localized in membrane protrusions. Thus, this study suggests that the 3' untranslated region of Rab13 mRNA plays a major role in the accumulation of Rab13 in membrane protrusions.
As seen, Rab proteins play a major role in vesicle trafficking and the progression of many forms of cancers, though its functions in these cancer pathways remains to be fully understood. Targeting Rab13 regulators and effectors may prove to be useful for future cancer therapies.